The process of DNA replication occurs within the S phase of interphase. It begins with a DNA molecule untwisting, which is done by the enzyme helicase. From here, the DNA molecule now resembles a ladder much more than its double helix shape. Replication will begin at specific sites called the origin of replication. Eukaryotic cells will have several origins of replication, while prokaryotic cells only have one. From here, the enzyme helicase will recognize this sequence and begin to unwind and separate the parent strands of DNA. As helicase separates the parent strands, single-stranded DNA-binding proteins will bind to the unpaired strands in order to prevent them from rejoining. Another enzyme called topoisomerase helps relieve the stress that is put on the rest of the DNA that hasn't been unwound. Now that the DNA has been completely separated, there are two strands, a leading strand and a lagging strand. The leading strand is oriented in the 5' to 3' position, while the lagging strand is oriented in a 3' to 5' position. Replication will begin with the leading strand. The enzyme primase begins by putting down an RNA primer that is complementary to the DNA strand. DNA polymerase III binds the RNA primer to the leading strand and begins adding DNA nucleotides. It moves in a 5' to 3' direction. Finally, in order to complete the leading strand, the RNA primer is replaced with DNA nucleotides. Each nucleotide that is added to the strand is composed of a deoxyribose sugar attached to a base and one phosphate group. The leading strand is completed when the enzyme ligase binds the last DNA nucleotides. Synthesis to the lagging strand differs because DNA polymerase III can only add DNA nucleotides to the 3' end and move down towards the 5' end. Because of this, the DNA nucleotides for the lagging strand must be added in pieces. Before this, however, primase will make and put down several RNA primers. DNA polymerase III will bind the primer on the 5' end. It will then synthesize a complementary strand that is much smaller than the continuous strand found in the leading strand. This is called an Okazaki fragment. Now DNA polymerase III moves on to the next RNA primer to bind it and synthesize the other Okazaki fragment. The RNA primers are now replaced with DNA nucleotides by DNA polymerase I. Now these nucleotides must be joined together with the Okazaki fragments, which is accomplished with the enzyme ligase. That completes the lagging strand, and the organism is now left with two complete identical copies of the parent DNA. The model in this stop motion has depicted DNA synthesis occurring flawlessly. However, this is not always the case. Errors will occur, but there are many proofreading mechanisms designed to prevent these from becoming imprinted into the final copy of the DNA, which would cause mutations. DNA polymerase II is responsible for such proofreading. It will proofread each nucleotide against its template, and if it finds a mismatch, it will remove this incorrect sequence and resume the synthesis correctly.